Modifications of chain terminator sequencing. As I told you that this Sanger technique of chain termination, it was developed by Sanger and co-worker in 1977, and then different modifications were introduced by different workers. As I told you that in case of Sanger method, the bases or the fragments, they were labeled with P35 phosphate, and then later on this radioactive material was replaced with another isotopes like sulfur 32 and even S35 sulfur isotope was also used in order to enhance the resolution of the auto radiograph. So different techniques were introduced. It is not only the radioactive material that one can use for the detection of DNA fragments in the process of auto radiography. इसके अलावा कुछ जो नॉन आइसोटोपिक मटेरियल हैं इनको भी यूज़ किया जा सकता है डीएनए की फ्रैगमेंट्स को डिटेक्ट करने के लिए, लेकिन जो ज़्यादा सूटेबिलिटी बनती है वो रेडियो आइसोटोपिक मटेरियल्स की है. एक तो ये बेसिक मॉडिफिकेशन थी जो सांगर का जो बेसिक मेथड था उसमें कि फॉस्फेट की जगह S32 या S35 रेडियो आइसोटोप यूज़ किए गए, तो इससे जो जेल की जो रेजोल्यूशन है वो इससे बढ़ जाती है. Another modification that was introduced in the basic method was the use of different type of DNA polymerases in place of clinofragment of DNA polymerase that is isolated from E. coli. And in this case, the DNA polymerase that was mostly preferred was T7 DNA polymerase. It has the same efficiency to introduce modified dideoxynucleoside triphosphates in the growing chain of nucleic acids. And another advantage of the use of T7 DNA polymerase is that its processivity it is high. That is, it can add more number of nucleotides in the growing chain of nucleic acid before its dissociation. So, this is one of the advantage of T7 DNA polymerase that replace the clinofragment of DNA polymerase. In addition to that, another DNA polymerase that was isolated from a bacterium which is thermophilic bacterium. ऐसा माइक्रोब जो कि ऐसे इन्वायरमेंट में ग्रो करता है जिसका टेम्परेचर रिलेटिवली हाई होता है और इसको कहते हैं thermophilic microbes like Thermus aquaticus. So this is the type of bacterium from where another type of DNA polymerase was isolated, and it was Taq DNA polymerase. And especially it is useful for those sequencing processes that are carried out at high temperature. So this was another modification that was introduced in the Sanger basic method. Another point about the Taq DNA polymerase is that if alanine base or phenylalanine it is replaced with tyrosine, then this Taq DNA polymerase it cannot distinguish between normal deoxynucleoside triphosphate or dideoxynucleoside triphosphate. So distinction or discrimination it is lost as a result of this minor changes in the sequence of the amino acid of Taq DNA polymerase. So as I told you that this technique, that is chain termination technique, it can be used in conjunction with M13MP vector, where we can get single-stranded DNA template for the sequencing purpose. And if this method it is exploited with S35 then it can also increase the efficiency of the sequencing. Later on, another modification was introduced where double-stranded DNA fragments can be used for the sequencing purpose. And here, the double-stranded DNA during the sequencing process, it can be denatured by using alkali or high temperature, and then later on it can be neutralized, and then by using specific primer it can be sequenced. तो हम ये कह सकते हैं कि जब डबल स्ट्रैंडेड डीएनए जो है वो सीक्वेंसिंग के लिए यूज़ किया जा सकता है, तो जो दूसरे वेक्टर हैं जिनकी रिक्वायरमेंट होती है कि वो सिंगल स्ट्रैंडेड डीएनए टेम्पलेट प्रोवाइड करें, लाइक M13 MP जो सीरीज है वेक्टर्स की, फिर उसकी ज़रूरत नहीं रह जाती. So these were the different modifications that were introduced by different workers during the course of time.